Hi, my name's Alexis Witt, and this is going to be my final presentation. In this, I'll be presenting Chapter 12, which is Biologic Oxidation. Biologic oxidation is the core of how life powers itself. This presentation will touch on topics such as energy metabolism, enzyme function, detoxification, and so much more. Let's talk about biologic importance of biologic oxidation. What is it? In the simplest of terms, it's when molecules swap electrons. So one molecule can lose electrons, that would be oxidation, and the other can gain electrons, and that would be reduction. This electron movement fuels many systems, including cellular respiration, ATP production, and detoxification. Without these processes, we as humans couldn't survive. If we look closely at the image below, you can see oxygen molecules interacting with substrates. A reminder that biological oxidation drives ATP synthesis. As a reminder, oxidation is when electrons are lost and reduction is when electrons are gained. These redox reactions are how cells breathe, eat, and work. A fun fact is some biologic reactions do not require energy. For example, dehydration reactions can transfer electrons without oxygen. Continuing on with biological importance, on this slide we will discuss the role of oxygen. In biologic oxidation, it is the star of the show and the final electron acceptor in many reactions. It allows for production of ATP. In the diagram on the side, which I'll show you right here, shows how oxygen acts at the end of the electron transport chain. And a fun fact, oxygen therapy can actually save lives. In respiratory failure, giving patients extra oxygen restores their ability to carry out cellular respiration and make ATP. Here I have my very first question, and I'll get it a little closer to the screen. It reads, what is the role of oxygen in biologic oxidation? So for option A, it states it's a structural element in enzymes. Option B, it's an electron donor in all reactions. In C, it says it acts as the final electron acceptor in many oxidation reactions, enabling ATP production and detoxification. D, it binds directly to glucose during glycolysis. Now, after reviewing the last few slides, the answer is most definitely option C. It acts as the final electron acceptor in many oxidation reactions. For my next slide, we will discuss free energy and redox potential. Every oxidation-reduction reaction has an energy change known as delta G. This will tell us whether the reaction will release energy or if we need energy input in it. We can also predict electron flow using the redox potential, which is symbolized here. Right here. Now, going back, the chart on the side helps us to visualize which reactions are spontaneous. A negative delta G means energy is released, which is a key idea in metabolism. Redox potential compares how likely something is to donate or accept electrons. It's measured relative to the hydrogen electrode at a pH of 7, which has a standard value of negative 0.42. This image shows a table ranking biologic redox systems by their redox potential values. It lets us predict the direction of electron flow. So, for my second question, the redox potential is commonly measured relative to which standard, and what is the value at a pH of 7? So, we can look at hydrogen electrodes at a pH of 0 with a value of 0 volts. Hydrogen electrode at a pH of 7 with a value of 0 volts. Option C reads hydrogen electrode at pH of 7 with a value of negative 0.42 volts. And option D reads hydrogen electrode at pH of 7 with a value of plus 0.42 volts. As in the last slide, the answer is going to be C, hydrogen electrode at a pH of 7 with a value of negative 0.42 volts. Now, for my favorite component of all of Chapter 12, we're going to discuss classes of oxidoreductases. It's funny because I have a very hard time pronouncing that. There are four main classes of oxidoreductases. We have oxidase, dehydrogenases, hydroperoxidases, and oxygenases. These oxidoreductases are like a crew managing an electron highway. So these oxidases use oxygen as a hydrogen acceptor. What that means is they take hydrogen from a molecule and they give it to oxygen. So this result will be water or hydrogen peroxide. For example, cytochrome oxidase is the final enzyme in the electron transport chain. So if cyanide were to block it, it would be deadly. I will show you. So this diagram here, it does show that oxidase is removing hydrogen and it hands it off to oxygen, creating water or hydrogen peroxide. It also shows cytochrome oxidase, which is essential for breathing. Now, in this next slide, we'll discuss dehydrogenases. Those do not require oxygen directly, so they will transfer hydrogen between molecules, often using coenzymes like NAD+, NADP+, or FAD. Seen in action in glycolysis, fatty acid synthesis, and the citric acid cycle. Here below, or on the side, I'll just show you a little closer. You can see that NAD+ and FAD accept hydrogen and electrons. These coenzymes carry energy from one place to another, and they let redox reactions happen without direct oxygen, such as in glycolysis. On my next slide, we'll talk about hydroperoxidases, and they act as sort of a cleanup crew. Peroxides and catalase destroy harmful peroxides like H2O2 before they can damage cells. So below, I put on peroxidase and catalase as sort of visual representations just to show that catalase in action breaks down hydrogen peroxide and it turns it into harmless water and oxygen. Peroxidase uses vitamin C, cytochrome C, either or, to reduce peroxides. This protects us from oxidative stress. Now, on the next slide, oxygenases are the oxygen incorporators. So these enzymes, they add oxygen directly to molecules. Dioxygenases insert both oxygen atoms, hence the di. And mono-oxygenases, so mono meaning one, like cytochrome P450, insert one oxygen and reduce the other to water. Now, I do have a question. So, for question 3, it does read, which enzyme is involved in reducing hydrogen peroxide, so H2O2, to water and oxygen in cells? So, for option A, it reads dehydrogenase, B is cytochrome P450, C is xanthine oxidase, and D reads catalase. So, after reviewing those last couple of slides, the answer is very clearly catalase. So, for my next slide, we'll be discussing cytochrome P450. So cytochrome P450 is a molecular multi-tool. So it's found in the liver, the intestine, and mitochondria, and it helps metabolize drugs and make steroids. So an interesting fact about cytochrome P450 is you metabolize medicine differently based on your genes. So your unique cytochrome P450 enzyme, it affects how quickly and slowly you can break down drugs, which I thought was so cool. Now, I do have another question. Hitting it back to back. So, for question 4, it reads, what is the primary function of cytochrome P450 enzymes in the human body? Option A reads, breaking down glucose in glycolysis. B is transferring electrons to NAD+ in the electron transport chain. C is metabolizing drugs and steroid synthesis. And D reads, converting oxygen into water during oxidative phosphorylation. So after my slide, it is very clearly C, drug metabolism and steroid synthesis. So lastly, One of the body's greatest threats is superoxide anion. It's a free radical that causes oxidative stress, aging, and even disease. That's where superoxide dismutase steps in, and it turns superoxide into a harmless oxygen and hydrogen peroxide. Now, a fun fact about superoxide and free radicals. The DNA damage and proteins contributing to all of these is gray hair, wrinkles, and even neurodegenerative diseases such as Alzheimer's. On my next slide, I'm going to discuss more on superoxide dismutase. Superoxide dismutase is found in all aerobic cells. Overall, its job is to jump into action before there's too much damage done. And a fun fact about this is plants, this is my baby plant I'm doing growing, plants need superoxide dismutase, and they need it to survive under intense sunlight, which mine is in my window. It increases oxygen reactivity and can damage their chloroplasts if not controlled. Now, for my fifth and final question, it is going to be, what is the function of superoxide dismutase in cells? So option A reads to oxidize fatty acids, B to synthesize ATP from ADP, C to convert superoxide anion into hydrogen peroxide and oxygen, or D initiates the citric acid cycle. So the answer is going to be C, to convert superoxide anion into hydrogen peroxide and oxygen. Lastly, to summarize Chapter 12, biologic oxidation powers metabolism and detoxification. The 4 oxidoreductase classes are as follows: oxidases, which use oxygen, dehydrogenases, which transfer hydrogen, hydroperoxidases, which defend against radicals, and oxygenases, which incorporate oxygen. Enzymes like cytochrome P450 and SOD are biochemical superheroes. Thank you for watching my presentation, and please refer to the following citations as a credit to this presentation. Thank you.